بسم الله الرحمن الرحيم. Welcome again, and today's video is the second one about LL-37 in NETOSIS. NETOSIS, as I explained in other videos, is a program for the formation of neutrophil or extracellular tracts or NETs that consist of modified chromatin decorated with bactericidal proteins that came from the granules and the cytoplasm of neutrophils. In NETOSIS, neutrophils arrive at the site of inflammation and are stimulated to release NETs and go into the process of NETOSIS. And these NETs are consisting of double-stranded DNA associated with histones and numerous other granule components like proteases and myeloperoxidases. LL-37 peptide also is released during this process and binds to NET chromatin or diffuses from the site beyond it. One of the important characters of LL-37 is its affinity for phospholipid membranes. So the binding of LL-37 to cellular membranes will compromise cellular integrity, and this is the basis of its antimicrobial potency. So it's not surprising that LL-37 contributes to nuclear membrane disruption and formation of NETs, helping the process of NETOSIS. When the NETs are released, LL-37 associates with the nuclear chromatin fibers, and due to its positive charge, LL-37 decorates the NETs, but the association with chromatin will reduce its antimicrobial activity. So association of LL-37 with the chromatin will decrease its antimicrobial activity. On the other side, NETs coated with LL-37 are resistant to bacterial nuclease degradation and thus may be more active in preventing microbial dispersion. As an example, psoriasis is a skin disease with prominent neutrophilic infiltration, and NETOSIS plays a role in its pathogenesis. We have a high local concentration of LL-37 and plasmacytoid dendritic cells are supposed to exacerbate the disease following stimulation by LL-37 in complex with DNA or RNA. So in psoriasis, LL-37 in combination with DNA or RNA may contribute to self-sustaining, amplifying vicious cycle of ongoing and progressive chronic inflammation. Lupus erythematosus is another example where LL-37 peptide-containing complexes protects the NET-derived DNA from degradation, will lead to accumulation of immunogenic NETs and induction of anti-NET antibodies. So pathogenic autoantibodies, including antibodies to LL-37, occur in patients with systemic lupus erythematosus. LL-37 antibodies may contribute to immune complexes forming LL-37 DNA anti-DNA complexes and further raise the production of NETs. This will lead to amplification of a vicious inflammatory cycle. Also, LL-37 peptide-containing complexes are potent stimulators of plasmacytoid dendritic cells, leading to type 1 interferon production. Also, they activate the memory B cells, and also LL-37 plays a critical role via activation of macrophages and inflammasomes and production of interleukin-1 beta and interleukin-18, and further amplifies self-propagating inflammation and causing lupus flare. RNA may be associated with LL-37, not only from self but also from foreign, like bacterial, viral, fungal, and exert immunostimulatory effects. An example of that is what's happening with coronavirus disease. So LL-37 can enhance toll-like receptor 3 signaling by interacting directly with double-strand RNA and enhancing the type 1 interferon response. Also, it can form complexes with single-strand RNA as it does with single-strand DNA, enhancing toll-like receptor 7, 8, and 9 signaling, which play an important role in systemic lupus erythematosus. So LL-37 can have effects in other autoimmune and inflammatory diseases through neutralizing lipopolysaccharide and altering the membrane integrity, stimulation of migration of cells of the innate immune system, affecting dendrocytes maturation, stimulated production of cytokines and chemokines, and through interaction with their receptors and triggering mast cell degranulation. LL-37 also caught the attention in the field of altered fibrosis, for example, in pulmonary disease, in which they found that LL-37 is highly expressed in small airway epithelium from patients with chronic obstructive pulmonary disease. A decreased production of LL-37 from cathelicidin HCAP18 in neutrophils has important consequences. This is a rare autosomal recessive disease caused by mutations in gene encoding lysosomal cathepsin C. For example, patients with Papillon-Lefèvre syndrome. In this syndrome, neutrophils of the patients lack or exhibit reduced levels of several types of proteases, including proteinase 3. The lack of proteinase 3 and deficient processing of HCAP18 in neutrophils may play a role in the exposure or the increased incidence of periodontal diseases in patients with Papillon-Lefèvre syndrome. As a final comment, LL-37 is characterized by structural flexibility, and depending on the ligands to which LL-37 associates, its structure changes and it cannot be followed upon the same criteria of detection. Actually, LL-37 belongs to a very large family of proteins better known as intrinsically disordered proteins or peptides. These intrinsically disordered proteins are often composed of modules with flexible bridges between the modules. Intrinsically disordered proteins show pleiotropic functions upon the partner they bind to. And examples of this, not only LL-37, we have other examples like P53, tumor suppressor gene, and HSPA8 protein. I hope that you have enjoyed the video, and I'm reminding you again to subscribe to the channel and activating the alarm bell if you are interested in getting any updates. Thank you.